Hi students, welcome back. So things have changed a bit since we were last in contact, and so I've decided in response to change up our lectures a little bit. So what we're going to do for our first lecture is just talk a little bit about viruses, what they are, how to kill them, and how one develops vaccines. And then we'll also talk a little bit about epidemiology and immunity, not in this clip, but in the next clip. Okay, so where do we start? Everyone has now heard of course, COVID-19 and the pandemic that's going on globally. How do we put that in a broader context of what we know biologically? So we know there are a variety of different microorganisms out in the world, both bacteria and viruses. Some that we are familiar with from prior to this incident are, of course, the flu virus, influenza, HIV, other coronaviruses before this novel coronavirus. So what are these actually? Well, if we think about viruses, they come in two different forms. They come in what's called an enveloped form and then also a naked form. And the picture you see over on the right is maybe a picture that kind of seems familiar at this point, since we've now had the spiked proteins of the COVID-19 virus kind of everywhere in the media, right? So it is an enveloped virus. So it has, as you can see here, this lipid membrane around the outside of it, and then these spiky proteins. So it's one of those. It's in that group. And I'm just going to focus on those in this lecture because of the current issues associated with what is actually known as the SARS-CoV-2 virus. Now, when I say SARS-CoV-2, you may not be as familiar with that terminology. You've probably seen from the UIS website and everywhere else is talking about COVID-19, or sometimes people will say COVID-19 virus. So what's up with all this terminology? Well, when we think about viruses, there's the virus itself, and then there's the disease that it causes. And we distinguish these two things, and they're given different names. So let's think of one that caused the AIDS epidemic, for example. So if we think of the virus itself, that would be the human immunodeficiency virus, so HIV, right? As opposed to the symptoms that it causes, the disease that it causes is AIDS. So if we think to today, that's where SARS-CoV-2 comes in. So this novel coronavirus, its official name is SARS-CoV-2, and the disease that it causes is the respiratory disease that unfortunately we've seen becoming more and more widespread globally, and that is COVID-19. Now, I do want to make a note here that there are lots of different types of coronaviruses. So if you are talking about this new one, it's important to say the novel coronavirus. So what is this little thing that has brought down, basically our country to its knees in many ways, right? So we're all, or many of us, are in isolation. The economy has really kind of crashed. All sorts of various things. It's this little, you would think, innocuous piece of biology. And so what a virus is basically a little bit of genetic information, and that can be either DNA or RNA, and then it's all wrapped up. Okay, so in the case of the novel coronavirus, it's an RNA virus. And so that just means that it's got a little bit of RNA tucked inside the middle of it, right? And then it's got, again, those proteins and that lipid membrane around it. So here's our definition: DNA or RNA surrounded by a protein capsid that replicates only inside host cells. So this is an important thing that makes viruses different from other kind of microbes that are out there in the world. So we know about bacteria. We've already talked about them in this class in terms of soil bacteria and gut bacteria, right? And they have everything they kind of need inside themselves. Yes, they're only a single cell, but they've got all that cellular machinery. Viruses don't work that way. So viruses don't have all of that cellular machinery. They're just a little wrapped up piece of genetics. So they actually are more dependent on their host than a bacteria is. They need to basically get that little piece of genetics into the host cell so that more viruses can be made. So as I've said in the second bullet here, they basically insert themselves into a cell, and then they can hijack it in a way and make that cell into a virus factory. So this figure is for influenza virus. You can see in the upper left, this is a cell, a host cell being infected by the influenza virus. So you see first the virus attached to the outside of the cell, get into the cell, so the second phase here. And then once it's within the cell, it opens up and releases its contents. Then in this case, the viral RNA goes into the cell's nucleus, and you can see in step 5, the nucleus is taken over, if you will, and starts to just produce virus particles instead of whatever that cell typically produced. And then what you see in stage 6 here is that the cell is still alive, right? So if you don't know enough or you don't know a lot about viruses, you might think that the virus kills the cell, but it doesn't do that. It just takes it over and makes it into a little virus factory. So the cell is still alive, but now instead of doing what it typically does, it's just producing lots and lots and lots of these viruses. We're most familiar, of course, with the viruses that are associated with human disease. And so I've put this table in. We've already talked a little bit about AIDS and HIV, right? And then you can just go down chickenpox, Ebola, hepatitis B, etc. And you can see all the way almost to the bottom, the disease is SARS, and then we have coronavirus. Of course, this is talking about the historic coronaviruses, right? Not the novel coronavirus which just arose in late 2019, early 2020. But what you can see is it is related to these others that are on here under SARS, and you can see again that structure that probably you've seen in the news. Oh, and I should also say with the SARS, you can see that the disease is similar in terms of a respiratory infection that these viruses cause. So, important things. How the heck do we kill these things, right? So, people are increasingly worried about viruses being on surfaces, about viruses being transmitted. There's been runs on hand sanitizer at the local grocery store. Why is that? How does that work kind of scientifically? Well, the reason sanitizers work is they use alcohol. So you can see in the upper left of this infographic ethanol, propanol, and isopropanol. So if you've got a hand sanitizer, look at the back. You can see it might have ethanol in it. It might have isopropanol. And what those alcohols do is they basically they denature the proteins on the outside of the virus. Now, you may not be familiar with that term, denature. It basically means to basically unfold the whole protein so that it can't work anymore, right? So kind of disrupt it. Even better than hand sanitizer for killing a virus is to wash your hands. Okay, so we've all been washing our hands 20 seconds, all these little parts, there's funny videos out there, all of that. Okay, so why does that work so well? It works that so well because soap is this really cool molecule. So if you look at the upper right, these soap molecules, they kind of look like little sperms, but they are not. They are little soap molecules, but they have the head on them, right? The hydrophilic head means that it really likes water. Okay, so one part of the soap molecule really likes water. Its tail, hydrophobic tail, doesn't like water. And so because of that, how soaps work is they basically pull things apart, right, which you can see in the lower left, because of that difference in kind of charge. So you can see the soap down in the lower left destroying the virus by because of the nature of that molecule pulling all of those little pieces of membrane in the virus, of protein in the virus, apart. And then the other beautiful thing about washing your hands is the water, right? So all of these little things, if there's one that maybe didn't get burst open, it will get flushed with the water down into the drain. So that's kind of the chemistry, if you will, chemistry, biology of why washing is so effective in killing these things. So we know for like the flu or for other viruses that are out there, we have vaccines developed, right? And so every year, when it comes to be flu season, people go and they get their flu vaccine, and then that helps give them some immunity for the upcoming season and the upcoming strains. So obviously, there's people have been asking, well, here's this horrible virus that is doing all of these things to our economy and to our lives. What? How do we get a vaccine for it? So we need to start with just thinking about, well, what is a vaccine? How do you make a vaccine? In this case, particularly for a virus. So what a vaccine does is we've talked about all those little spiky proteins and all those little receptor proteins, all those proteins on the outside of the virus, right? So your immune system, that's partly what it will trigger into in terms of its whole way of killing things, right? So what you can do in a vaccine is you can just isolate that protein, and you can make a vaccine with just those are called coat proteins, just those coat proteins. And there is, of course, variation in vaccines. So sometimes in particularly older vaccines, they would just be killed virus, right? So it is the virus, but it's been killed, so it's just little parts of it. Okay, so you have this coat protein, you've made a lot of it, okay, and then of course if it's gone through all these stages that we'll talk about a little bit, you have the vaccine. Now, when you give that vaccine to people, that will allow those people to develop immunity, right? So these viral proteins, then those people have an immune response. And then when the virus comes, their immune system's already kind of primed, if you will, to fight the virus very, very quickly. And so then they don't often exhibit the worst signs of the disease. So why does this take so long? Why can't we just, boom, come on, scientists, make a vaccine? Well, it takes a long period of time for a couple of things. So we've got the virus over on the left. You can see I talked about the receptor binding domain, right? So that's the receptor protein that we want to try to get the immune system to recognize, right? So you need to isolate that, right? And then you can see current stage development of vaccine candidates, right? So there's a lot of different approaches, as I've said, that you can use to come up with candidate vaccines, including live attenuated vaccines, all the way down to just having those recombinant proteins, etc. So you make all of those various candidate vaccines, but then of course you have to see, will it really work? So is it effective? When you give this to people, do they actually develop the antibodies that they need to make, and are those antibodies really useful in killing the virus, right? And then of course effective is part of what we want, but we also want it to be safe, right? So when we put this recombinant protein into somebody, do they get sick? Do they get, so all of those things. So both safety and efficacy. And then, of course, the final stage is, as we've already talked about in this class, there's billions of people on the earth, right? So how do you ramp up, assuming you have something that, yes, it's effective and yes, it's safe, how are you going to make so much of it that you can vaccinate, in this case, billions of people? Okay, that's it for this snippet. I'm sure you still have questions. We can talk about those in the discussion board. I do want to give you kind of a sneak peek to the next snippet, where we'll be talking more about immunology and also epidemiology. So people might be curious, why can this virus spread more quickly than something like a flu virus, or something which we have vaccines for, or even some other virus that isn't novel. This gets back to immune systems. So you see in this graphic, for herd immunity, you have on the top sustained transmission of the virus, and then at the bottom you have terminated transmission. So what you can see at the top, okay, you've got this little green guy and he's sick. So that's the transmitting case, right? Now, if this is a new virus, it's out there, all of the folks that this sick person may encounter are susceptible. They don't have immunity because this is a whole new virus, and no one has been exposed to it before, right? So you have susceptible, then that person becomes a transmitting case, then that next encounters the next susceptible person, and this gets into these issues of exponential population growth, etc. So you have this sustained transmission. As opposed to if you do have a vaccine, or even if you have a virus that is not new, so the population has some level of immunity against it, right? In which case, then you have transmitting case A, right? And then that person's immune, so it didn't get transferred to that person, the yellow person here, right? And so then it doesn't get to another susceptible individual. Okay, so we'll be talking more about those dynamics in the next little snippet. I miss seeing you all face to face. Stay safe, wash your hands, and we'll talk to you soon. Thanks.